All right, well, we're continuing on with chapter 27, which is on metabolism. I'm looking at the schedule. Today is Monday the 10th. We have 4 more class periods, and the syllabus plans for chapters 28, 29, and 30. Some semesters we don't get to chapters 29 and 30. And so I'm going to just sort of play it flexible over the next few lectures. I think we'll finish most of metabolism today. We will definitely get to chapter 28. Don't stress out if you find that study guides for 29 and 30 are not posted and there aren't any problems in the problem set on 29 and 30. We will get to as much of that as we can, but we're going to just play it by ear. Some of you may not be comfortable with that. That's okay. You don't have to be, because it's my class, right? Anyway, I will try to keep you sort of posted as to what to expect. You can definitely expect chapter 28. 28 is back to chemistry. We're going to talk about basically transition metal catalyzed reactions in modern organic synthesis. It's going to be unlike anything you've ever seen before in terms of reactivity, and it's really outside my area of expertise, so we'll be learning together. Chapter 29 is really kind of fun, and we could spend a lot of time there. It's on reactions kind of like the Diels-Alder reactions, but also some photochemical reactions. If we get there, we'll talk about why you need sunlight to make vitamin D. And then chapter 30 would be on polymers. You've mostly gotten that in the chapter on radicals, but it might be nice to come back to it again. So we're just going to see. But for now, you know, chapters 27 and 28 will basically be as scheduled. We'll finish 27 on metabolism today or next time. Probably next time also we'll move into chapter 28. Other housekeeping items, you do have an exam that closes today. I know that roughly 25% of you've taken it, so the remaining 75%, make sure you get it done and watch that testing center closing time. Some of you have emailed me because we usually have study guides and practice exams due when the exam closes, just as a flexible deadline. I made sure that those study guides and practice exams are due today when the exam closes. Hopefully you've been doing them all along, but this gives us some schedule flexibility. Okay, so we had gotten to step 3 of glycolysis. This is an investment phase. Let's see, I need a new note document. In which we have invested another molecule of ATP. Remember that we're converting basically a phosphoanhydride bond within ATP, and if you don't remember the structure of that, go look it up in your notes from the previous class, and converting that into a phosphoester bond with the C1 hydroxyl of fructose 6-phosphate. We talked a lot about fructose last time. I told you this step was irreversible. It's quite downhill in energy. And this is the second molecule of ATP that we have consumed in order to get the process going. You'll see what the phosphate groups are for a little bit later. In fact, it's these phosphate groups that we're going to use to generate new molecules of ATP a little bit later on. Okay, so in step 4, we're actually going to break the fructose 1,6-bisphosphate into 2 3-carbon pieces. And the enzyme that does this is called aldolase, which tells you what the reaction is going to be. It's going to be in one direction an aldol reaction, and in the other direction a retroaldol reaction. This reaction's very reversible. I, of course, don't want you to memorize delta G numbers. I'm simply providing them for context. You can see that the equilibrium constant here, if delta G is near 0, the equilibrium constant is close to 1. And you can see that that's the case here. So it's fine to name this for the forwards or the backwards reaction. The products, the way we're showing the reaction, aren't really that much more or less stable than the starting materials anyway. You'll notice that fructose has 6 carbons in it, and we're going to get 2 molecules out here. One of them is going to be a 3-carbon piece that looks like this. This portion corresponds to carbons 4, 5, and 6 of fructose 1,6-bisphosphate. So, I mean, now they should be numbered 1, 2, and 3, but I'm just going to keep the numbers on there so you can see where the molecule comes from. This one is called glyceraldehyde 3-phosphate. And then the other portion corresponds to carbons 1, 2, and 3 of fructose 1,6-bisphosphate. We'll use, I don't know, pink for those 3 carbons. And even though now they're separate molecules, we should number them differently. We'll just call it that. This 3-carbon piece is called dihydroxy acetone phosphate. All right, and notice that this one is an aldehyde, this one's a ketone. It's not immediately apparent if you look at this cyclic hemiacetal form of fructose bisphosphate what's going on here, but it will become clearer if we open up the ring. So you should be able to draw the mechanism for hydrolysis or opening up of a cyclic hemiacetal. I'm not going to show you that here, but if you go back to our earlier chapter on aldehydes and ketones and look up the mechanism for acetal formation and acetal hydrolysis, this will become clearer to you. We've talked about it already, so I'm just going to gloss over it now. In the ring-opened form, carbon 2, which I don't like how that looks, carbon 2 is a ketone, and the oxygen on carbon 6 is an alcohol. Some of you have asked about this, but notice that when opening to the open chain form, none of the other stereocenters in the molecule change at all. The only thing that changes is the anomeric carbon, which becomes a ketone or an aldehyde, and you just break the bond between the OR group and the hemiacetal carbon. The oxygen from the OH group becomes the oxygen of the ketone or the aldehyde. So stay with me here. You have a carbonyl carbon, an alpha carbon, and a beta carbon. This is a beta hydroxy carbonyl compound, which is an aldol product. So what we need to do to get to those 2 3-carbon pieces is a retroaldol reaction. Now remember, in the aldol reaction, an enolate attacks the carbonyl carbon to make a new bond between the alpha carbon of the enolate and the carbonyl carbon of the electrophile. So the bond that we need to break in the retroaldol reaction is the bond between the alpha and the beta carbons. And we need to just think backwards. The final step of the aldol reaction to give the beta hydroxy alcohol is the tetrahedral intermediate picks up a proton. So the first step of the retroaldol reaction is deprotonate the beta hydroxy group. Oops. All right, so now we have our negatively charged beta hydroxy group. The next step of the retroaldol reaction is for electrons on the oxygen to kick down and form a new carbon-oxygen pi bond, and to break the carbon-carbon sigma bond between alpha and beta carbons, with the electrons going out onto the alpha carbon. That's going to take us back to our aldehyde electrophile and the enolate nucleophile. For just the sake of consistency, I'm drawing the molecule in the same basic orientation that we had it in the full-length molecule before we broke the bonds. So that's carbons 4, 5, and 6. This is our aldehyde portion. And then carbon 3, 2, and 1 are here. This is the enolate. And then, of course, getting to the final product, dihydroxyacetone phosphate, is as simple as picking up a proton from some source, the enzyme or solvent, and that gives you glyceraldehyde 3-phosphate, and it gives you dihydroxyacetone phosphate. That's the retroaldol reaction. Questions about that? Yeah.
Why is it called a dihydroxyacetone when there's only one hydroxy group on it?
Well, it's named as a derivative. Yeah, why is it called dihydroxyacetone phosphate? It's named as a derivative of that, which is dihydroxyacetone, and then you'd put a phosphoester on one of the OHs. Yeah, that's all. Okay, other questions? If you're looking backwards at where we've been, the fact that we did a retroaldol reaction should explain to you why we bothered to do this isomerase step, where we moved the carbon-oxygen pi bond from carbon 1 to carbon 2. Notice that in glucose 6-phosphate, the carbonyl carbon is on carbon 1. Glucose 6-phosphate is likewise a beta hydroxy carbonyl compound, but if we did the retroaldol reaction at this stage, that would clip glucose into a 2-carbon piece and a 4-carbon piece, and then we would have to have different sets of enzymes to deal with those two pieces. That's tremendously inefficient. Nature compensates for that by moving the carbonyl over through this isomerization, 2 tautomerization steps. Now when you get to fructose, the beta hydroxycarbonyl compound, the retroaldol splits you into 2 3-carbon pieces. And we're going to see in the next steps, we're going to actually convert dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. And then we're going to use identical machinery from that point on to do the reactions. So this is all about nature arranging things so that it can funnel both 3-carbon portions down the same pathway. Why reinvent a whole set of other mechanisms when you can use what's already available? All right. So that's, from this point on, actually, I told you this was the last investment step. From this point on, we're going to be doing basically payoff steps where ultimately we're going to be setting up to generate ATP. Step 5 is going to be sort of an off-pathway one. This is going to be one where we convert dihydroxyacetone phosphate into glyceraldehyde phosphate. The mechanism of this, you will see, is basically, 5, identical conceptually to what we saw before when we converted glucose 6-phosphate into fructose 6-phosphate. If you compare dihydroxyacetone phosphate with glyceraldehyde phosphate, it's clear that what we need to do now is move a carbon-oxygen pi bond over one carbon. The mechanism or the reaction that does this is, the enzyme that does this is called triose phosphate isomerase. If you ever get into or learn a lot about biochemistry, you'll learn about what are called Michaelis-Menten constants, and which describe sort of how efficient an enzyme is. Triose phosphate isomerase is one of the most efficient enzymes. It's almost what we call diffusion limited, meaning the enzyme makes the reaction go so fast that the thing that limits the rate of the reaction is simply the substrate diffusing through water to find the enzyme. So it's one of the best enzymes out there. Even so, enzymes don't change the free energy change of the reaction. This reaction is totally reversible. So now I got to get rid of the blue numbers and get rid of the shading, because all we're basically doing, and maybe I should draw dihydroxyacetone phosphate, just pancake flip it and draw it this way. At this point, we're going to change carbon 2, which is a ketone, into an alcohol, and change carbon 1, which is an alcohol, into an aldehyde. The mechanism, I'm going to just say C step, whatever it was, from glycolysis, C the glucose 6-phosphase isomerase step. The mechanism is going to be exactly the same. You're going to do this via tautomerization, and there should be an ene diol intermediate. I'll draw that ene diol intermediate, and then you ought to go back and make sure you can figure out how to do this mechanism. It simply involves proton transfers and resonance structures of enolates. So it shouldn't be too hard, but this is the ene diol intermediate, and I'll let you fill in the details from there. All right, anything you want to ask about that? Yeah, go ahead.
Last time you didn't label a step as step 2, like step 1, step 3.
Oh yeah? Sorry. I'm sorry, that sounded kind of rude. I mean, maybe I put it there after you said that to cover my sins, but yeah, this is the isomerase of glucose 6-phosphate to fructose 6-phosphate is step 2 of glycolysis. Yep. Others? Okay, so now everything we do from here on, you have to remember we're doing twice, because we have 2 equivalents of glyceraldehyde 3-phosphate. Here is the first one, and here is the second one. That's going to be important in keeping track of whether we've actually paid off the debt that we incurred by consuming 2 molecules of ATP for every molecule of glucose. So I, of course, won't write the reactions twice or draw the mechanisms twice, but everything from 6 through 10, you have to remember happens 2 times. Okay, so starting material for 6 is glyceraldehyde 3-phosphate. We're in the end game now. our goal is to recover the 2 molecules of ATP that we have consumed and to get other energy-rich molecules out that we can use for other purposes. So this is going to involve some changing around of functional groups. Ultimately, glycolysis is going to end in a 3-carbon molecule called pyruvate. So we're not going to break any more carbon-carbon bonds in this process, but we are going to move bonds around. So if you look at some of the differences here, carbon 1 in pyruvate is a carboxylic acid, so that tells you we're going to be doing some redox chemistry. Carbon 2 is a ketone. Here it's an alcohol, so that suggests a couple of different oxidation steps. And then we notice that the phosphate and the OH group are just gone completely. And that we're going to have to address as well. But our goal, as I said, is going to be to recoup the 2 molecules of ATP and also to generate some additional molecules of ATP. So let's show you step 6. Step 6 involves an oxidation. We're going to use NAD+, and we're going to get NADH out. That's considered payoff because we can take NADH to the electron transport chain and exchange it in mitochondria and exchange it for ATP. We also need some phosphate, which we can draw in the protonation state that it would mostly be in at pH 7. Your biochemistry textbook will probably call this P with a subscripted I. That stands for inorganic phosphate. That just means a conjugate base of phosphoric acid that's not attached to a carbon. The end product, you'll see that this is an oxidation because we've oxidized from an aldehyde not to a carboxylic acid, but to something that's actually even more useful. And that is basically a mixed phosphoanhydride. You can almost see the anhydride. If this were a carbon, this would look exactly like an anhydride. And if you want to have a sense for what nature's doing with this reaction, we're setting ourselves up to generate another molecule of ATP because this portion of the molecule is now a carboxylate, which in some biochemical reactions can be a decent leaving group. All right, so let's show you how this happens. It's thought that phosphate acts as a nucleophile to attack the carbonyl carbon to give you a tetrahedral intermediate. Oh, and I should say this reaction is also reversible. I'm writing down delta G's not so you can memorize them, but rather so you can just see the difference between reactions that are very, very downhill versus reactions that aren't. Here's the tetrahedral intermediate. Oops, I'm missing an oxygen. So that's your negatively charged oxygen. Here's your hydrogen. And here's the phosphate group. Again, the pKa of the proton here is about 7, so at physiological pH, about 50% of the molecule is protonated here, and about 50% is its conjugate base. I may show it either way. Don't worry about that. The oxidizing agent at this stage is NAD+, which we're going to draw. And you've seen before. I'm going to draw it in this resonance structure. You could draw it in the other resonance structure form. This one requires fewer arrows, so it's a little bit more efficient. Electrons on the oxygen kick down. Hydride is removed and handed directly off to the para carbon, the carbon that's para to the nitrogen. And let's see, that's it. And if you want, I'll draw the structure of NADH for you. Some of you are asking, where did the proton go? And I already told you not to worry about it. It's removed by water because the pKa of that proton is around 7. Could be 50% off, 50% on. Doesn't particularly matter. Okay, questions about that? Yes?
Is the last step reversible as well?
Yes. Yeah, the delta G is for the overall reaction, so yeah. The enzyme that does this is called glyceraldehyde 3-phosphate. It's a big long name. Here's the name of the substrate, or what the enzyme acts on, glyceraldehyde 3-phosphate, and then what the enzyme does, dehydrogenase. No surprises there, because you see that NAD+ is part of this. Sometimes this enzyme is abbreviated as GAPDH. If you are ever in a molecular biology lab, cell biology lab, biochem lab, sometimes you will use GAPDH as an internal standard or an internal control in your Western blots to verify that you've loaded about the same amount of protein in each well in a SDS page gel. If that doesn't make sense to you, don't worry about it. You may never need it, but you will, if you're in a lab where you're doing Western blots, you may encounter people talking about this enzyme. Okay. So you can sort of see in this final material what nature is trying to do. I told you about getting a phosphate on something that's a good leaving group. And I should point out that this product is called 1,3, that's for 1, 2, and 3, bisphosphoglycerate. The bis means there's 2 phosphate groups, one on carbon 1, one on carbon 3. Why is it not di? I don't know. I've never really figured out when to use bis and when to use di. There's probably a rule for that. But if I don't know it, you certainly don't need to. Glycerate refers to the oxidized form of this aldehyde. So if this were a carboxylic acid, instead of being glyceraldehyde, that's now glycerate. Okay. Other questions or concerns about that? All right, step 7 is an example of what biochemists call substrate level phosphorylation of ATP. That means we're getting ATP by a direct reaction of ADP with an enzyme substrate. in contrast to the kind of ATP, or in contrast to the way we get ATP from the electron transport chain, where we're driving a proton gradient, and you'll learn about that in biochem. So if you ever hear that, excuse me, term, substrate level phosphorylation, they're talking about a reaction where ATP is directly generated by a reaction of ADP with the enzyme substrate. What does substrate mean? That means the molecule that the enzyme acts on. So the substrate for this reaction is 1,3-bis-phosphoglycerate. And I want to make sure I don't get this wrong. This reaction is also neutral, thermoneutral, meaning the delta G is basically 0, and it's therefore reversible. The reaction has ADP react directly with 1,3-bisphosphoglycerate to get ATP out. And then your product afterwards is just that leaving group that I told you about. Right? So all of this portion of the molecule is the good leaving group. And the fact that this reaction is thermoneutral, even though you're generating ATP, is impressive. And the reason for that is that you're basically taking this unstable mixed phosphoanhydride from 1,3-phosphoglycerate, and then you're turning it into another kind of phosphoanhydride in ATP. So trading one relatively unstable anhydride for another isn't such a big deal. The mechanism for this reaction is going to be very similar to the reactions you've seen before that have been catalyzed by kinases. And indeed, the mechanism here, the enzyme here is called phosphoglycerate kinase. It is actually named for the backwards reaction. The product of the reaction is called 3-phosphoglycerate. Glycerate refers to the carboxylic acid version of glyceraldehyde, and then the phospho on the the carboC3 hydroxyl is where the 3-phospho comes from. Mechanism is going to be basically the same as we've seen before for kinases. Here is the structure of ADP. I'm going to just use the base for A. ADP is going to have 2 phosphate groups on the 5' hydroxyl. And we're going to use one of those oxygens on the beta phosphate group as a nucleophile. That's going to attack the phosphorus at the C1 phosphorus on 1,3-bisphosphoglycerate. We're going to show the same basic mechanism that we've shown before for kinase type reactions. That is a pentavalent phosphorus intermediate. I'm going to abbreviate the rest of ADP like that. Actually, I hate that. I don't know why, but just got to go with my gut. And then over here, we have that phosphoglycerate leaving group that we've talked about. All right. Then electrons on one of the phosphorus oxygens will kick down and kick off the leaving group. And that gives you your product plus ATP. All right? So that pays our debt. Remember, everything we're doing here is twice. So we invested 2 molecules of ATP. We got 2 molecules out. We've now broken even. Of course, if that's where we stopped, then this has all been a fun exercise in making bad investments. But we've got more to do, because if you look at where we're trying to get, which is pyruvate, we've accomplished the oxidation of C1 to a carboxylate. We've still got some oxidation to do here, and we've still got to get rid of this phosphate group. Okay. And I guess I should mention, and maybe it's worth highlighting in I don't know, some kind of fiery color that where all the payoff is. NADH we can take to the electron transport chain, and in a little bit we'll discuss about how many ATP molecules you can get there from every molecule of NADH. But we've got one of those, and then, yeah, we've paid off our initial investment of ATP in 2 additional molecules of ATP there. Okay, questions about that? I probably won't show kinase steps like this in the future, simply because they all basically proceed through a similar mechanism. You've seen it once, you can continue forward from that. Okay, step 8 is going to look pointless, and we're actually not going to learn the mechanism of step 8, not because it's unknown, it is totally known, but I decided the juice wasn't worth the squeeze. There's a complication in terms of what an enzyme side chain is doing, and oops, didn't want to get rid of that, I just wanted to copy it. So we could talk about it in a lot of detail. It's not really worth it, so we just won't. This is another reversible step. The free energy change is again near 0 in cells. The step actually moves the phosphate group from the C3 hydroxyl to the C2 hydroxyl. That ends up being pretty important for setting us up for subsequent chemistry, but it's simple enough conceptually that we're not going to worry about the mechanism. The enzyme that does this is called phosphoglycerate, and then this has kind of a funny name, mutase. Not generating a mutant, but that's just moving or change, I think. But it moves the phosphate group from the 3' hydroxyl to from the C3 hydroxyl to the C2 hydroxyl. This is now called 2-phosphoglycerate. Okay, and we'll just pause to notice this is a highly charged molecule. You should expect there to be some charge repulsion between the carboxylate and the phosphate group. Again, it's possible and even probable that one of these OHs, one of these negatively charged oxygens is present as the conjugate acid. We won't worry about that. A lot of these enzymes are magnesium dependent, and so it's possible there's also a counter ion in the form of magnesium that would attenuate how bad it is to have 2 negative charges close to each other. So don't worry about the mechanism on step 8. Step 9, and we're almost there, again, looking where we need to be. We want to get to pyruvate, and we also want to make another equivalent of ATP. So in step 9, we're going to take 2-phosphoglycerate, and this is basically just going to be proton transfer. We're going to convert 2-phosphoglycerate into the following molecule, which if you've heard about glycolysis before, is called phosphoenol pyruvate. It's the enol of pyruvate. If we were to draw, I drew pyruvate above, I'll redraw it here. Here's pyruvate. This is the enol of pyruvate, and you put a phosphate on that OH group, and you have phosphoenol pyruvate. Again, this is a reversible, basically thermoneutral step. In cells, basically not a huge change in terms of free energy from starting materials to products. The enzyme that does this, because you've got the word enol in the name of the product, this is called enolase. And the mechanism is basically an E1CB type dehydration of something that is basically a beta hydroxycarbonyl compound. So we're basically going to do a dehydration step. Conceptually, you can think of this like a dehydration step after an aldol reaction. We're going to remove the alpha proton first with some base, probably from the enzyme active site. That's going to generate an enolate intermediate. Why are we doing things that way? Well, we need to eliminate OH, and OH minus is a terrible leaving group. So it makes sense that we should have to first make the enol. And then once we have a negatively charged oxygen, then electrons can kick down and the OH can leave. And that gets you to the phosphoenol pyruvate product. Okay? So the theme so far in glycolysis is most of these steps are reactions that you've seen before. Biology is using tautomerization and carbonyl chemistry to accomplish its purposes. All right, anything you want to ask about that step? Okay, let's do the last one. In the last step, we're going to generate 2 molecules of ATP, again, because this step happens twice. Step 10 is another example of substrate level phosphorylation. This is the final irreversible step of, and I don't have the number written down, so I can't demonstrate how irreversible it is, but I'll just write down that it's irreversible. The final irreversible step of glycolysis, it's quite downhill in energy for reasons that we'll talk about. Here's phosphoenol pyruvate. There's your starting material. Dang. I was looking in my notes and suddenly I don't have an enzyme name. I've been so good about that so far. So, oh, I do know what it is. If you forget what an enzyme name is, you can sometimes help yourself remembering it. Help yourself remember it by thinking about the name of the substrate, either the starting material, and then what the enzyme does. This converts ADP back to ATP. So it could be named for the product substrate and the backwards reaction, which is phosphate transfer. This enzyme is called pyruvate kinase. But it's irreversible. What's going to happen in this reaction mechanistically is we're going to do a kinase step, which is going to involve transfer of a phosphate from the C2 hydroxyl phosphoenol pyruvate to the beta phosphate group of ADP. So in terms of kinase steps, that part's going to be familiar. And then the leaving group, if we draw phosphoenol pyruvate a little bit better, you can see that the leaving group here if that left and picked up a proton on the oxygen, that would make the enol of pyruvate. And of course, tautomerization from the enol to the more stable keto form is going to be downhill in energy. So that tautomerization helps pay for some the transfer of the phosphate group from here to ATP. All right, so let's sort of draw that mechanism. I'm going to abbreviate the ATP structure. To do that, I'm just going to draw the beta phosphate group of ADP. And then here I'm just going to put AMP, so that's adenosine monophosphate. What I'm not drawing is the alpha phosphate, the ribose ring or the adenine ring. And hopefully that's okay. We'll have lone pair electrons on the phosphate oxygen attack this phosphorus. We'll have, as we've done before, a penta coordinate or pentavalent phosphorus intermediate. Likely this is magnesium dependent to help deal with all of the negative charge that's there. Again, that's the beta phosphate group of ADP. I need to draw the rest of pyruvate. It's thought that electrons on the oxygen, one of the phosphate oxygens, kick back down in carbonyl style elimination. And it's thought that the leaving group picks up a proton and that protonation on the oxygen is probably faster. You might be thinking, couldn't that leave as an enolate and then just pick up a proton on the alpha carbon and get us straight to pyruvate? It's thought that protonation on the oxygen is faster, and then you get rapid, after that, tautomerization from the enol to the keto form. And there, again, carbon-oxygen pi bonds are more stable than carbon-carbon pi bonds, which helps pay for the whole process. And I'll just write ATP here because we just made ATP, and again, that's another example of substrate level phosphorylation. Okay, so looking at where we began, or rather, before we do that, any questions about the pyruvate kinase step? Okay, good. So going back to where we began, this was our overview of glycolysis. We actually made 4 molecules of ATP, but we're subtracting the 2 that we invested in those first 3 steps. And we made 2 molecules of NADH. It's thought that you can get roughly 2 to 3 molecules of ATP from the electron transport chain for every NADH that you make. So you've made somewhere between, let's see, can I do simple math? No, I cannot. Somewhere between 4 and 6 molecules of ATP here. In addition to that, it's about 8, okay, from 1 molecule of glucose. And that's anaerobic respiration. And if you're a prokaryote like a bacteria, like a little E. coli, this is how you survive. Of course, We're eukaryotes. Our cells have nuclei. In addition to cytosol, we also have mitochondria. And mitochondria are able to take NADH and exchange it for ATP. I guess with prokaryotes, you just have to survive on this ATP and use the NADH for other stuff. In any case, there's a lot of oxidation and ATP generation left to be done. If you look at this pyruvate, there are 3 carbons there still. And you all know that you breathe in oxygen and you breathe out CO2. The CO2 that you breathe out comes from these 3 carbons. So there's more chemistry to be done. We're going to want to somehow oxidize each of these 3 carbons, a carboxylate group, a ketone, and a methyl group, all individually into CO2. That's kind of complicated, it's challenging, and it turns out that you need not only enzymes to do this, but you need an existing pool of other substrate molecules that we can incorporate pyruvate into and then move functional groups around to set ourselves up to lose CO2. Preview, newsflash, you want to lose CO2, you guys already know how to do that. That's through what's called decarboxylation. So you're going to want to be thinking in the next phase, how do I set myself up to do decarboxylation reactions? We've always done that so far with beta keto acids, and we're going to continue to do that. If you look at pyruvate, though, we got a problem because pyruvate is not a beta keto acid, it's an alpha keto acid. So nature has a whole elaborate set of machinery that's going to facilitate doing stuff with alpha keto acids to do decarboxylation. So decarboxylation is the name of the game for what we're going to do next, the next metabolic pathway, which is called the citric acid cycle. But that's a good place to pause. So let's take 5, and then when we come back, we'll dive into that portion of metabolism.